Welcome back to my channel Knowledge Chronicle,
where we unravel the mysteries of science in the most engaging way possible. Today, we're diving deep into one of the most fascinating aspects of biology, enzymes. These microscopic powerhouses drive nearly every chemical reaction in living organisms. From digesting food to copying DNA, enzymes make life possible. But how do they work? What makes them so special? And why are they so incredibly efficient? Stay with us till the end to find out. What are enzymes? Enzymes are biological catalysts, special molecules that speed up chemical reactions inside living organisms. Almost all enzymes are proteins, though some special nucleic acids called ribozymes also act as catalysts. To understand enzymes better, let's first recall how proteins are structured. Proteins are made of long chains of amino acids, folded into intricate shapes. Enzymes also follow this pattern. They have a primary structure, amino acid sequence, a secondary structure, folding into alpha helices or beta sheets, and a tertiary structure, a complex three-dimensional shape. But the most crucial part of an enzyme is its active site, a small pocket or groove in which a specific substrate, reactant, fits perfectly like a key into a lock. This is where the magic happens. How do enzymes work? To truly understand enzymes, let's break down a simple chemical reaction. Chemical reactions, the basics. Chemical compounds undergo two types of changes. 1. Physical changes. These involve a change in shape, state, or phase without breaking molecular bonds. For example, ice melting into water. 2. Chemical reactions. These involve breaking old chemical bonds and forming new ones. For example, barium hydroxide plus sulfuric acid gives barium sulfate and water. This reaction forms new substances, making it a chemical transformation. Enzymes lower the energy required for such reactions, making them happen much faster than they would on their own. For example, the enzyme carbonic anhydrase speeds up this reaction. Carbon dioxide plus water gives carbonic acid. Without the enzyme, only 200 molecules of carbonic acid form per hour. With the enzyme, this number skyrockets to 600,000 per second. This insane speed up by 10 million times is what makes enzymes so incredible. Factors affecting enzyme activity. Enzyme activity depends on several factors, including temperature, pH, and substrate concentration. 1. Temperature. Enzymes work best at an optimum temperature, often around 37 degrees Celsius in humans. Below this, they become slow and inactive. Above this, they can denature, lose shape and function. However, thermophilic enzymes from bacteria in hot springs can survive at 80 to 90 degrees Celsius. 2. pH levels. Each enzyme has an optimum pH. For example, pepsin, a stomach enzyme, works best at pH 2, while amylase, a saliva enzyme, prefers pH 7. Extreme pH changes can denature enzymes. 3. Substrate concentration. Increasing substrate concentration increases enzyme activity up to a limit. Beyond this saturation point, all enzymes are occupied, and the reaction rate levels off. Enzyme inhibition. How reactions are controlled. Sometimes enzymes need to be turned off. This happens through enzyme inhibition. 1. Competitive inhibition. A mimic molecule competes with the substrate for the active site. For example, malonate inhibits succinic dehydrogenase, stopping metabolism. 2. Non-competitive inhibition. An inhibitor binds somewhere else on the enzyme, changing its shape so the substrate can't bind. Classification of enzymes. Enzymes are grouped into 6 classes based on their function. 1. Oxidoreductases. These transfer electrons in oxidation-reduction reactions. An example would be dehydrogenases. 2. Transferases. These move chemical groups from one molecule to another. For instance, kinases. 3. Hydrolases. They break bonds using water. A good example is lipase. 4. Lyases. These remove or add groups without using water. Think of decarboxylase. 5. Isomerases. They convert molecules into isomers which have the same formula but a different structure, like isomerase. 6. Ligases. These join two molecules using ATP. A classic example is DNA ligase. Cofactors, the hidden helpers. Many enzymes need extra molecules to work. These are called cofactors and are divided into 3 types. 1. Prosthetic groups. These are permanently attached organic molecules. For example, heme in catalase. 2. Coenzymes. These are temporary helpers derived from vitamins. An example is NAD+ from vitamin B3. 3. Metal ions. Some enzymes need metals like zinc or magnesium to work. For instance, carboxypeptidase needs zinc. If the cofactor is removed, the enzyme stops working, proving its essential role. So, to summarize, enzymes are biological catalysts that speed up chemical reactions. They work through the enzyme-substrate complex model. Temperature, pH, and substrate concentration affect their efficiency. Enzymes can be inhibited to control reactions. They are classified into 6 major groups. Many enzymes require cofactors for activation. Enzymes are truly nature's molecular machines, performing complex tasks with amazing speed and precision. Without them, life as we know it wouldn't exist. If you enjoyed this deep dive into the world of enzymes, don't forget to like, share, and subscribe to Knowledge Chronicle. Your support helps us create more valuable content for you. Stay curious, stay informed, and we'll see you in the next video.